Today we are going to talk about the role of medullary collecting duct in urine formation. So far we have discussed the function of each and every part of the nephron, like proximal tubule, loop of Henle, distal tubule, and cortical collecting duct. And we have talked about their role in the urine formation, the maintenance of body fluids, and maintenance of acid-base balance in the body. Similarly, the last portion of the nephron, the medullary collecting duct, it also has a very important role in urine formation. Now, medullary collecting duct normally will reabsorb less than 10% of water and sodium. It also has its role in the reabsorption of urea and secretion of hydrogen, and it has important role in the final formation of the urine. Now, they are known as medullary collecting duct because they are present in the medulla. The cells in the medullary collecting duct are cuboidal with less mitochondria and less metabolic activity. Now, we discussed that less than 10% of water and sodium is reabsorbed in the medullary collecting duct, and the reabsorption of water from this tubule here into the medullary interstitium is with the help of ADH, antidiuretic hormone. As we discussed previously in the cortical collecting tubule, that reabsorption of water is dependent on antidiuretic hormone, and antidiuretic hormone release is dependent upon the volume of water or the volume of blood. It's dependent upon the fluid level in the body. So whenever there is a high need of conserving the fluid, there is loss of fluid, then a lot of antidiuretic hormone is secreted from the pituitary gland, and this antidiuretic hormone acts apart from other areas of the nephron on the medullary collecting duct. It means if there is ADH secretion, there will be reabsorption of water from the medullary collecting duct as well. Now, another important thing is that there is some reabsorption of urea as well from the medullary collecting ducts. We discussed that there is no reabsorption of urea in this segment, the cortical collecting tubules, but there is slight reabsorption of urea from the medullary collecting duct into the medullary interstitium. Now, the interstitium present here near the medulla is the medullary interstitium. As we discussed, for example, these are the nephrons. All nephrons are not at the same level. And when the nephron starts, it starts from the proximal tubule, the loop of Henle, the distal tubule, and then the cortical collecting duct. Similarly, this nephron has all these components, but the collecting ducts are either cortical collecting tubule or the medullary collecting ducts. So the difference is that these cortical collecting tubules, they are present in the cortex of the kidney. They are present in the cortex of the kidney, the outer portion of the kidney, while the medullary collecting ducts, they are basically present in the medullary portion. So this line basically shows that the collecting ducts present above this line are present in the cortex, and the collecting ducts below this line, they are present in the medulla, and they are known as the medullary collecting duct. So the urea reabsorbed in the medullary collecting duct basically stays in the medullary interstitium. When a lot of urea starts reabsorbing into the medullary interstitium, the overall osmolality of this region increases. The osmolality of this region increases. Now, this high osmolality of this region helps in the formation of concentrated urine. We will discuss this thing in detail in our coming lectures, but the urea reabsorbed from the medullary collecting duct stays in the medullary interstitium and overall increases the osmolality of the medullary interstitium, and the high osmolality of the medullary interstitium helps in the formation of concentrated urine. Finally, in the medullary collecting duct, apart from reabsorption of water with the help of ADH and reabsorption of some urea, there is secretion of hydrogen. There is secretion of hydrogen into the tubule. There is secretion of hydrogen into the tubule. Rather than reabsorption, there is secretion. As we discussed, that there is secretion of hydrogen in the proximal tubule, and this secretion of hydrogen is not very much against the very high gradient. Similarly, there is secretion of hydrogen in the cortical collecting tubule, which is basically secretion of hydrogen against a very high gradient. If their secretion is against the gradient of around 1 to 10, here in the cortical collecting tubule, we discussed that the secretion is against a gradient of as high as 1:1000. Similarly, the secretion of hydrogen from these tubules, from the cells of the medullary collecting ducts into the tubule, is also against a very high gradient. It is also against a very high gradient, and the secretion of hydrogen means the hydrogen will be secreted into the tubule, and it will be going along with the urine outside the body. And this secretion of hydrogen now helps in the regulation of acid-base balance of the human body. So medullary collecting duct is helping in conserving the fluid by the reabsorption of water with the help of ADH. It is helping in increasing the osmolality of the medullary interstitium, and finally it is helping in the acid-base balance by secretion of hydrogen. So that's all about the functions of different parts of the nephron. Thanks a lot for watching the video.